Hey guys, and welcome to our exam review for micro. So in this review, I'm going to briefly go over some of the key points on our study guide and then point out where we can find the answer and then get you thinking about what types of questions I can ask about those certain concepts. So just a disclaimer, this review video is not like 100%, it's more or less just a mini comprehensive of what is fair game for the exam. So let's go ahead and get started. So in chapter 1, we discussed scientific names of genus and species. So with that, I definitely want you to know what does a proper organism name looks like when written. So let's go ahead and go to that chapter 1 so I can show you. So in that, just gotta find the slide. Okay, so the proper way of actually writing a scientific name for an organism is that you will have the genus capitalized. So in this case, Staphylococcus is the genus, and then you will have the species is capitalized, and then the genus is all lowercase. And then whenever it's written, it's in italics. So this is the proper way to write the name of an organism. Or you can abbreviate it, such as E. coli down here. So what you'll do is take the first letter of their name and then period, and then write out their last. So write up their species name. So that is how you'll properly do that. So that is how and what I would ask in terms of that concept. Okay, so with that, let's go back. So the three domains and classifications and the big differences between eukaryotes and prokaryotes. So with that, I definitely want you to know the three domains of life. So the three domains of life is bacteria, archaea, and eukarya. So all of these eventually branched out over time, essentially, and they all have a common ancestor. So I definitely want you to know the fact that bacteria, archaea, and eukarya, we all have a common ancestor way, way back millennia ago, like millions of years ago. We do come from the same organism, but then we branched out over time. We diverged from each other into these three domains of life. And then some major differences between prokaryotes and eukaryotes are as follows. So bacteria and archaea, these are the guys that are considered prokaryotes. With that, they are very, very small, and then also their cellular structure is very different. So with these guys, they have something called cell walls, whereas eukaryotes do not. General eukaryotes do not, meaning like mammalian and like humans, but plants and some fungal cells have cell walls. So just because you're a prokaryote or a eukaryote doesn't mean you don't have cell walls. Mammalian cells don't have cell walls though. So that would not be a significant difference. Now, an actual key difference between prokaryotes and eukaryotes is the fact that how they organize their cell is different. So with bacteria cells, they don't have any membrane organelles inside their cells. All they have is just certain cell parts in order to function. Whereas eukaryotic cells, they are very different. So let me actually just pull up a picture real quick so I can show you. And just go to Google's. So as we see right here, there is not a lot of cell parts in bacteria as compared to eukarya. So the true characteristic has to do with the absence of membrane-bound organelles. So prokaryotes do not have that, whereas eukaryotes, we do. So that's the main thing. They both have DNA. Certain eukarya also have cell walls, and then they both have proteins in them for cellular function. Okay. Okay, so with that, let's talk about these two dead guys real quick. So we have Robert Hooke, and then we have Anton van Leeuwenhoek. I believe that's how you say it. So Robert Hooke, he was the first one to ever build a compound microscope. He was the one who actually saw the cell first. He did not observe bacterial cells, though. That was Anton. Anton's the one who observed bacterial cells, whereas Robert Hooke, he looked at more plant cells. He looked at the cork of a tree. So this is actually what he saw. This is what he drew. So he's the one who termed the cell, yeah. We'll leave it at that. That's kind of a poor sentence, but that's okay. So he was the one who actually first saw it, whereas Anton, he was the one who actually observed a bacteria cell. But at this point, whenever he was observing bacteria cells, microbes and everything was not linked to disease yet. But it will be soon. So that's the main difference that I want you to know between them. Yes, that's the main difference. Okay, so with that, let's go ahead and go to our next concept. So we're going to talk about biogenesis and spontaneous generation. Okay, so with this, I want you to know what is spontaneous generation. So spontaneous generation is the idea that living microbes just come to be. Like all of a sudden they're there and we don't know why. But as we know, that is not true. And spontaneous generation was disproved by Louis Pasteur. And the reason why it was disproved was because he created this swan neck flask that allowed air to come in but didn't allow debris or like any contamination to get in. So with that, he was just like, okay, organisms, living organisms, they need air to survive, and he disproved that by allowing oxygen to come in, and he used the swan neck flask. So definitely know that. So know the idea of spontaneous generation, who disproved it, and how did he disprove it. Okay. Okay, so cautious postulates. So cautious postulates are as follows. So I definitely want you to know the 4 postulates and then be able to apply them to like a scenario. So if I was to give you a scenario on the exam, so I'll give you like a kind of like a case study, and then what I would do is that ask you cautious postulates in terms of which one is not relevant. So if you were to apply the scenario that I gave you to cautious postulates, which of the following choices would not be relevant to the case study? So I know it's kind of confusing, but so let me just give you an example real quick. So let's say that you have a elderly patient. He works with livestock and everything. He buried his livestock because they died of anthrax. Then the patient who was taking care of the dead bodies, he was wearing gloves and everything. He was moving them to the burial sites. But then 4 days later, the elderly man who was working with the diseased cows, he sees that he's starting to get sick. And then over time, he will seek medical attention. And then from that, the physician will observe him. He'll look at his like skin lesions and everything, and then the physician will prescribe some medicine, and then from there they will test, like they'll do a biopsy, and then they'll determine what is causing these lesions, and from there like they determine that the patient now has anthrax, and then the patient will continue medication and he will get better. So that is something somewhat of a case scenario that I may give you on the exam, and then from there I would want you to know the 4 cautious postulates and know which part of the cautious postulates would not be relevant. Okay, so let's go to the discovery of DNA. So The scientist that discovered DNA was Rosalind Franklin, so that's towards the bottom. So Rosalind Franklin, she was the one who actually discovered the structure of DNA using X-ray crystallography. So I definitely want you to know that. She was the pioneer behind discovering it. And then Watson and Crick, these were the scientists that actually published her work. Although Rosalind Franklin was not credited in their Nobel Prize winnings, but that's okay. So I'm mainly concerned about you knowing Rosalind Franklin and then the method of which she used to discover DNA. We'll go more into DNA throughout term about like how does it pair, how do we actually get the structure that she discovered. We'll do that in around chapter, I believe, 8 or 9. So as of right now, just know who discovered it and how, like what was her method. Okay. So with that, I believe the last thing that I want to talk about in terms of chapter 1 is endosymbiosis. So the idea of let me go one more slide back. So the idea of endosymbiosis is that organisms, you started off with a smaller organism and then a larger organism came along and engulfed the smaller one, and then they started living together as one. So it's a smaller organism living inside of a larger organism. So this is the figure that I do want you to definitely know. So the idea of endosymbiosis was that mitochondria, so mitochondria right here, so this is like pre-mitochondria, and then pre-chloroplast, which is cyanobacterium, they have their own genetic sequence, they have their own DNA. But then what happened is that along came some kind of protist and some kind of plant cell that uptook these pre-mitochondria and pre-chloroplast cells, starting living together as one organism, and then eventually became to what we know today as the modern plant cell, animal cell, fungal cell, and protist. So it's the idea that that these either cyanobacteria or these protobacteria was engulfed, and then these guys represent the pre-chloroplast and pre-mitochondria. Okay, so I believe that will wrap up chapter 1 for the most part. So let's go ahead and transition into chapter 2. So chapter 2, it says, what is the difference between pathogenesis and virulence? So let's go ahead and talk about pathogenicity and virulence. Okay, so first let's talk about pathogenicity. So this is the actual ability for a microbe to cause disease. And in order for a microbe to cause disease, it is dependent on numerous factors. These are just some of them on the slide. So depending on the genetics of the microbe can determine how does it interact with a host, and then also on which it can enter the body. So as we know, some microbes only like entering the body in one way. So if an organism only likes to enter the body in one specific way, such as, let's say, through the skin, then it may not be as pathogenic as other microbes that can use different ports of entry, such as like the skin, the nose, the eyes. So just because it only has one port of entry doesn't necessarily mean it's not going to be virulent. It just means that it has maybe not as many opportunities to enter a host. And then also, how does the pathogen interact with the host immune system? So if an organism can avoid the immune system, then generally speaking, it will be a very good pathogen. As compared to if our immune system can detect it very, very quickly, very easily, and get rid of it, then it is not a very good pathogen, because the whole idea behind being a pathogen is to cause disease. Okay, so let's talk about the definition of virulence real quick. So virulence is the measurement of how severe a disease will be. So there are certain organisms that are extremely virulent because they have such a high mortality rate, meaning that they will kill a good majority of their population that they infect. So, such as like Ebola, let's say a certain Ebola strand has a 90% mortality rate. That means that if there was 10 people infected, 9 of those people will die. So that means that it has high virulence. Whereas, let's say we have um, the common cold. So the common cold is not very virulent as compared to the Ebola, because let's say the common cold only has a 10% mortality rate, so it will only kill 1 out of every 10 people. So virulence has to do with how severe the disease is. And then next on our list was the lethal and infection dose. So I definitely want you to be able to interpret a graph such as this on the exam. So meaning that if I was to put a graph on the exam like this, the graph on the exam would show the percent mortality, just like this, how it shows, and then also as well as the dose. And in that, I would want you to tell me what is the lethal dose at, let's say, 50% for the second agent. So with that, I would want you to be able to read that, okay, so this line represents agent 1, whereas the orange line represents agent 2. So from there, I would want you to be like, okay, so according to this, agent 2 is at 600. That is pretty much the extent of which I would want you to do. Whereas if I ask for agent 1, then it would be 400. Now that's assuming if I was asking for 50%. Now let's say if I was asking for 75%, then you would go roughly around here. So 75% is maybe slightly above 700. And then for this one, it's probably around 450 or so. So that is how I would want you to interpret this table and be able to apply it to a question. Okay. Okie dokie. So let's talk about what actually makes a pathogen successful. So there are certain criteria that a pathogen needs to follow in order to be successful. And let me find that slide so we can look at it together. Here we go, sorry. Okay, so there are three things that need to be present in order for it to be successful. The pathogen has to be able to attach to a host cell in order to cause infection, has to be able to avoid the immune system to some certain extent, and then also be able to obtain nutrients. And then also, one that's not on here is their host range. So generally speaking, if you have a very narrow host range, you may not be as successful as compared to a broad host range. But that's not really true, because it's not accurate, because there are certain viruses that are only found in like humans, so such as like the human papillomavirus. That virus is very, very successful, but it has a narrow host range. It's only found in humans as of right now. So just because a pathogen has a narrow host range does not mean it won't be successful. It may mean that it may take longer to be successful, but it can still be successful, especially since HPV is a very, very, very common virus that we do find in society. So the fundamentals that have to be there, though, in order for a pathogen to be successful is attachment, immune avoidance, and be able to obtain nutrients. So meaning they have to be able to acquire it one way or another from the host. Okay. So with that, I think that's going to bring us into the differences between signs and symptoms. Okay. So the differences between signs and symptoms, let's pull up that slide, is that a sign is something that is objective, meaning that you can the physician can physically see it and measure it. So you can measure a fever, you can categorize a rash, whereas a symptom is specifically to what the patient is feeling. So you can't really measure pain very accurately, but you can still measure it because everybody has a different pain tolerance. And then, as well as malaise, which is like weakness or like discomfort, everybody has a different level on that. So some people can still function when they're extremely tired, whereas other people may not. Same thing with pain. So I know somebody who gave birth, and she, every single time they asked her about her pain, she's like, "Oh, I'm a 2." I know for a fact if I was giving birth, I would be like a million. So it just, that symptom varies between patients. So on the exam, what I will probably do is something like, I will give you a list of signs and symptoms, and in the question I'll be like, which of the following is a sign, or which of the following is a symptom? So I would want you to be able to tell the difference between that. And then next, we're going to talk about what is the difference between epidemic, pandemic, and endemic. So with these guys, I want you to be able to tell me, if I gave you a situation. So if I said, like with the coronavirus right now. So we have the coronavirus going around. As of right now, it has been documented in China, it has been documented stateside, and then I believe even on a cruise ship, they say that somebody tested positive on a cruise ship off the coast of, I want to say Japan, but I'm not too sure about that. But either way, with those guys, which what category would that fall into? Now, the most closely one, it would probably be a pandemic, although we are not there yet, but it may turn into a pandemic because it is an epidemic right now, since there has been a lot of cases of coronavirus, especially in China, that has spiked over a short period of time, such as, I believe it's been roughly 3 to 4 months since the first documentation of it. So as of right now, the coronavirus is categorized as an epidemic because there has been a spike of cases over a short period of time, but it has the potential of becoming a pandemic because it's starting to spread across the globe. So that is how I would want you to use this terminology in a problem. Okie dokie. So what's next? Okay, so certain reservoirs and modes of transmission. Okay, so let's talk about modes of transmission first. So modes of transmission, what I want you to know is the differences between direct and indirect. So just like before, if I gave you a scenario, I would want you to tell me, is it a direct or indirect? And then based on that, give me an actual like example of why it was direct and what makes it indirect. So let's say that Susie went hiking, and on her hike she got very thirsty, but then she realized that her water bottle was empty. She found this stream, and then she filled up her bottle from the stream. So based on that, is that indirect or direct? So it's indirect because she ingested contaminated water, and that is an example of a vehicle. Or it could be something like Susie has lice but doesn't know, and then she shares her brush with Lucy, and then Lucy gets lice from using her comb. So that would be an example of a fomite because the transmission factor was the brush, and the brush is an inanimate object. So that is roughly how I would want you to use all this information on an exam. Okay, and then, so that's modes of transmission. Let's talk about reservoirs. So a reservoir is where a pathogen normally resides within that environment, or within either within or on the animal naturally. So an example of that would be a tick. So a tick is a vector because there's a pathogen that naturally lives on the tick or inside the tick, but it's also a vector because the tick can transmit it to a person. And another example would be the EEE. So this is Eastern Equestrian Encephalitis. So the bird is the reservoir, meaning that that pathogen generally lives with the bird, it doesn't cause harm to the bird. But when the mosquito bites the bird, but then the mosquito bites a human, then that means that the mosquito transmitted it to the person, so making it a vector. And then a carrier is a specific special type of reservoir. This is where a person does have the potential causative agent of illness, but doesn't necessarily mean that they are sick. But just because that they're a carrier doesn't mean that they can't become affected later down the line. And then also with reservoirs, a lot of times they don't necessarily they're not presenting disease. So just because they're a reservoir doesn't mean that they have disease or will never have the disease. They have the potential of getting the disease, yes, but the likelihood is that they don't necessarily show that they are diseased. So kind of like with the whole swine flu, some pigs, they had it, but they were not exhibiting disease all the time. Same thing with the avian flu. Okay. Okay, so we kind of already briefly talked about broad and narrow host range. So broad means that it can infect multiple types of species, whereas narrow, it can only infect one species. And then also with number 14, I really just want you to know the idea that every microbe has a certain way that they like to get in, and then based on the microbe that you have in your body can also determine way of exit. So generally with humans, the big way of giving away or getting out certain microbes will depend on what do the microbe cause. So the most common ways is vomiting and diarrhea and coughing. Coughing and sneezing kind of go together. So those are the three main ways that a microbe will exit. But if you are actively getting sick, so like let's say you have let's say that you have a parasite. So you have a parasite. Now the parasite doesn't want to leave. So the preferred way of a parasite leaving in cases of you trying to get better is none. But if the parasite wants to continue to spread throughout the human population, his preferred exit might be through a mosquito bite. So the mosquito bite will suck up your blood with some parasite eggs in it and then go transmit it to another person. So it kind of just depends. But just know the idea that every organism has a preferred way of getting in and getting out of a body. Okay. So I believe that will wrap up chapter 2. So chapter 3, I'm just going to briefly go over this really quick because chapter 3 I don't really test too much on because we're actually going to get tested on it more in terms of lab. So I'm just going to point out the really key highlights that I do want you to know. So definitely the gram staining. So this slide, I really want you to know this concept forward and backwards because we are going to be using it a lot throughout the semester. So I definitely want you to know the process of gram staining and know that gram staining is an example of differential staining, meaning that you're trying to tell the difference between two different cell types. So I want you to know the process of which, like, you have to adhere cells to the slide. You're going to add crystal violet, and then you're going to add iodine in order to create a complex within the cell. So either the crystal violet will stay within the cell or it may not. And then from there, you're going to wash it off with some decolorizer. And then based on what cell type you have, whether it be a gram-positive or a gram-negative, will determine the color of the cell. So if you have a gram-positive, then if you put on decolorizer, that crystal violet stain will stay on. Whereas if you have a gram-negative, the decolorizer will wash off that crystal violet. And then from there, you need to counter stain so you can actually see the cell. So as we see right here, if we stopped at the decolorization stage with gram negative, you may not be able to see it under the microscope because they're going to be clear, or with lac color, I should say lac color. So you're going to counter stain with something called saffron, and then with saffron, if you have a gram positive, the saffron is not going to stick to the cell because the crystal violet is already there. Whereas with gram negative, saffron will stick to the cell and stain the gram negative cells reddish, kind of reddy orange. So I definitely want you to know that for sure. Okay. And then, give me 1 second, guys. Let me double check about the whole oil immersion question. Yes. So I also want you to know, why do we use oil immersion? So the whole reason why we use the oil immersion on a specimen is so that we can maintain the same amount of refractive light, so we can uniform that reflective light, reflected. Sorry guys, just getting over being sick and very, very tired. So reflected light. So the whole idea is to minimize or to uniform it from in between the slide to the lens. So we just want to make it nice and uniform so it doesn't bounce off everywhere, so we can actually get a very concentrated area of light on our specimen so we can view it. So it's just the whole idea is to uniform it, to uniform the refracted light, or to like maintain the same amount of reflective light. Oh my god, reflective light. Okay, so that's pretty much all I want you to know in terms of oil immersion. Also in this lecture, I believe I pointed out, yes, the basic differences between a gram-negative and gram-positive cell. So definitely know that, love that. If I were to put a picture of their cell wall, of the cell of a gram-negative or gram-positive, I would want to be able to tell me that this one's gram-negative, this one's gram-positive, based off of the descriptions below. So gram-negative only has one single layer of peptidoglycan, whereas gram-positives have multi-layer. But we'll see that again in chapter 5, if I'm not mistaken. Okay, so was there anything else for chapter 3? Magnification and resolution. Okay, so one last thing for chapter 3 before we go into chapter 5, excuse me, is the concept of magnification and resolution. So let me see. Okay, let me see what the question would be like. Let me look at some of my examples real quick, guys. Yeah, so really I would just want you to know the definitions between magnification and resolution. So resolution specifically has to do with you can pinpoint the distance between two separate objects. So in this case, so like right here, this is more resolved as compared to this one, because you cannot tell the difference between the two spots in this image, whereas this image you can. So resolution has to do with being able to find the smallest distance between objects, but yet still be able to tell that they are separate objects. Whereas magnification, where so whereas magnification, what it does is that it will increase the size of an image in order to increase resolution. So what you would do is that, as we saw in lab on our microscope lab, that as we increased magnification, the object got bigger, essentially. So that's pretty much the extent that I want you to know for that in terms of our study guide. So that was the big things from chapter 3. So let's go to chapter 5. So let's look at chapter 5. Oh, just kidding. Actually, let's talk about the macromolecule exercise real quick. So that answer key is posted. So definitely take a look at that. The big thing from that is that I want you to be able to tell me distinctly what are the 4 macromolecules and what makes them them. So like a carbohydrate, a carbohydrate is all sugar. Lipids is a glycerol and fatty acid chains. And then like a nucleic acid has to do with a sugar base, phosphate, and a nucleoside on it. And then what was the last one? Proteins. Proteins are made up of amino acids. So if I were to give you some figures or just ask you a question just like that, I'd want you to be able to tell me that this is a carb, or this is a nucleotide. Oh, excuse me, guys. And then also, like if I was to give you a lipid, I would want you to be able to tell me is it saturated or unsaturated. And then also the main differences between DNA and RNA. So there was that big table. I pointed out the big ones that I wanted you to know, such as the structure of them, in terms of if they're double-stranded or single-stranded. And then what kind of sugar base do they use? So DNA uses deoxyribose, whereas RNA only uses ribose, as well as their function. So the function of DNA is that DNA is specifically genetic material, it's our blueprint. And then RNA, what it does is that it will take that genetic information and turn it into proteins to make proteins. And then in terms of their location, so DNA is found in the nucleus only, whereas RNA can be found in the nucleus as well as floating around in the cell, in the cytoplasm. So that's really it that I wanted you to get from that organic molecule exercise. So let me show you real quick where you can find that key. So if you were to go to your ACES account and go to Canvas, and of course the internet wants to be slow. I guess I don't blame it. It's Sunday. So let me go ahead and get to our Canvas so I can show you real quick. Sorry guys, I don't know why it's taking forever. Okay, I'll show you at the end of the video. This is taking way too long and I have no patience. But you can find it under our announcements. So that's where I posted it. But I'll show you at the end of this video. Okay, so let's go ahead and just move on to chapter 5. So chapter 5, I definitely want you to know the difference between a gram-positive, gram-negative, and something called an acid-fast cell wall, which is the microbacterium. So definitely be able to tell me, like if I was to put these images on the exam, which one's a gram-positive, which one's a gram-negative, as well as what are some key components, like maybe like some key differences between them. So let's actually just go to that slide so I can list them all for you. So in terms of gram-positives, they have a very thick cell wall because they have multi-layers of peptidoglycan. Also, they have this, oh, sorry, my eyes are itchy. They have something called an S layer, which is this guy right here, whereas a gram-negative does not have that S layer. They have these lipoproteins. Okay, so that's really the key components of a gram-positive cell, is that they have multi-layers of peptidoglycan, whereas gram-negatives do not. And then also they have that S layer, and then also gram-negatives do not. Okay, so gram-negative cells. So with gram-negative cells, they have one single layer of peptidoglycan. They also have an outer membrane. So this outer membrane is made up of lipopolysaccharides. Just lipopolysaccharides. They have no S layer, like I said before. And then also they have something called these O polysaccharides. So this is just sugars, a lot of sugars that allow them not to be phagocytosed. So meaning that they can't be eaten up by our cells and killed, essentially. So it definitely aids in their what do you call them? Their virulence and their ability to cause disease. Okay, so those are just some key components. Also, our last one, which is the acid fast, are also known as microbacterium. So these guys, they are categorized as gram-positives because they only have, they have that multi-layer of a peptidoglycan, but they have this very, very complex outer layer, their capsule. So they have these extra layers that are not found in gram-positive cells. And then also what makes them very specific is this malicholic acid. So this malicholic acid is specific to microbacterium or specific to acid-fast cells. So definitely know that. So that's pretty much the extent of which I will ask for these guys as of right now. Later on in term, I'll definitely go more into detail. But as of right now, I just want you to know structurally how are they different, and I would want you to be able to tell the differences between an image, like if I was to give you these figures on the exam. Okay, DNA replication. Okay, so DNA replication. So bacterial cells do not have sex. What they do is that they just clone themselves. So I want you to know, where does the process begin? So the process begins at something called the origin of replication. And then what will happen is that eventually the cell will replicate its DNA. And then from there, it will, after it gets to this point of termination, so it'll replicate in both directions. So it'll replicate, go this direction, and then go to this direction. And then eventually, as it replicates, it will hit this termination sequence. Once it hits that termination sequence, it will undergo something called septation. So this is where it'll start cleaving inward on itself, so such as right here. So it's starting to form that septum. And then eventually it will separate all the way across, and you will have two separate cells. And then their DNA is not stored within a nucleus, so they have something called a nucleoid. This is where all their genetic information is located. And then the way that it's organized is something called supercoiling. So we'll see this later on, but supercoiling is just a way of condensing their DNA into one area. And that is as far as I want you to know at this point, but we will go a lot more into detail into the process of DNA replication as well as protein synthesis. Okay. Okie dokie. So I believe that will wrap up that concept. Okay, so some structures and their functions. So with bacteria, they have a lot of specialized structures that we do not have. So the big ones are like the pili, the fibri. These two are specific for attachment. They are very, very important to bacteria. If a bacteria does not have them, the likelihood of them being successful is definitely minimized. So I do want you to know what is the whole purpose of these guys. So the whole purpose of these guys is to attach. So it could be attachment to another host cell or to another cell or to a surface. So that's the whole purpose of them. Now, another structure that is very important to bacteria is something called a flagellum or flagella if you're going to talk about multiple. So there are bacteria that can quote unquote swim. So if a bacteria has flagella, they have better movement as compared to bacteria who do not have it. So if I was to ask you, okay, we have this bacteria, he has the ability to swim along a certain surface, what is the structure that is being used in order to swim or in order to aid in his movement? And I would want you to say flagellum. Okay. Those are the big guys that I want you to know. Yeah, those are the big guys. There are some other specialized structures that they do have. So, like, we do not have these structures. So, depending on I'm sorry, I had a total brain fart. So depending on what structure they have can also be dictated by their environment. So like what we talked about in class, like these gas vacuoles, so certain microbes that live in aquatic environments, they will probably have these gas vesicles, whereas compared to if a bacteria doesn't live in an aquatic environment, it probably won't have these gas vacuoles. But so structure can definitely be dictated by environment. So just keep that in mind when thinking of like scenarios. So if I was to describe an area or like an environment, I would want you to be able to tell me what structure would be advantageous for it based on that environment. So if I was to say that this is a sulfur-rich environment, and it is also in an aquatic environment, so the water has very high sulfur deposits, I would want you to be able to be like, okay, it might be good for them to have sulfur granules as well as the gas vacuoles. So that's the big thing that I would want you to be able to get from this, is to be able to tell me, based on this, what structures would be advantageous for that organism to have. Okay. So we've already kind of talked about the differences between prokaryotic and eukaryotic structures. Let me look through the slides real quick if there's anything else. Okay, so the big thing that I would want you to get from the last part of lecture 5 is to know those organelles or those cell parts and their function. So I would want you to be able to tell me that the whole purpose of a lysosome is to go around and digest anything that is not necessary and to process it for nutrition. So that's just an example of what I would want you to get from this. We'll definitely see these cell parts and organelles later on throughout the semester, so I do want you to know what is their job. So that is pretty much the last part of the lecture. So Like, I want you to know what the rough ER is, the smooth ER, lysosomes, as well as Golgi apparatus, the nucleus, as well as its parts, and then the difference between mitochondria and chloroplasts. So that's what I want you to get from the end of chapter 5. Okay. So, with that, let's go ahead and go into chapter 6 really quick. With chapter 6, I really just want you to be able to tell me the first differences. So, this table right here. So, I would want you, like, if I was to describe an organism, I would want you to tell me whether or not he is an organotroph, a lithotroph, phototroph, chemotroph, so on and so forth. And then maybe even if they are an anaerobic or aerobic organism, because we do talk about that later on in this lecture, where certain microbes prefer oxygen whereas other organisms do not. So I would want you to be able to tell me that. So from those definitions specifically, if I was to describe an organism saying the organism gets his energy from, let's say, oopsies, let me go to this one. So if an organism was to get his energy from an electron donor, so if it's an electron donor, then that automatically means he's a chemotroph of some kind. But specifically what kind of chemotroph? So you would have to be like, okay, so I would say that he gets it from like a inorganic material. So if it's inorganic, then it is a lithotroph. And then from that, I would state a certain environment that is either oxygen rich or oxygen lacking. And then you would say either organic, anaerobic or aerobic. That's pretty much as far as I'll ask. I really just want you to be able to apply these concepts to situations. That's pretty much the big theme of my test, is to make sure that you can actually use all this terminology and apply it to a situation. Okay. Okie dokie. So let's go ahead and move on to our next concepts. So the differences between certain medias. So that will be the same idea with this nutrition of a microbe. So if I was to describe a certain media to you, I would want you to be able to tell me sorry guys, where's that slide? So based on all of this information, I would want you to be able to put it and apply it to the question. So if I was to describe a media, I would want you to be able to tell me which one of these categories does that media fall into. Now remember, just because these are 4 different categories doesn't mean a media can't be an enriched and differential. So just remember that it can be both. So that's all I'm going to say about that. Different counting methods. So some of these we did not go over. I do definitely want you to know the one about optical density, which is the turbidity one. So I definitely want you to know this concept. So the overall idea of this concept is that the more light that can travel through your sample means that the less dense your sample is, meaning the least amount of cells. Whereas if less light travels through, the more dense your sample is. So that is what I want you to know primarily for cell counting, because I did not go into hemocytometer or the FACS counting system. I really focused on the turbidity because we're actually going to see that in lab, so I care more about this one. So definitely know the concept of turbidity and optical density. Number 5 on the study guide, don't even stress about it because we are not going to do that. We're not going to calculate generation time. But in terms of actually knowing for the generation of cells, I do want you to know their growth cycle. So know this figure, love this figure, be able to explain this figure to me, and be able to tell me what is going on in each stage of their life or their growth. Excuse me. So oh my god, sorry guys. There are 4 phases. There's the lag, there's the log, stationary, and death. And I want you to be able to tell me which part of the graph is each, and then also what is happening in each phase. So that's all I'm going to say about that. And then lastly, just some ways to limit microbial growth, and then as well as biofilm and sporulation. So with that, let's go into what a biofilm is real quick. So with biofilms, they are a kind of like a community of either one bacteria or multiple organisms. It can be one, or it can be more than one organism. It is a multi-layered surface, so it's not just one layer of microbe, it is multiple layers of it. And like I said, it can be associated with either one species or multiple species. It can be either all bacteria, it can be all fungal, or it can be bacterial and fungal. It's kind of like a little bit of everything. The most common example that you'll see is probably plaque, or that you've heard of, excuse me, is plaque on the teeth. So just because there is a biofilm present does not necessarily mean it's associated with disease or an infection. But having a biofilm, though, can make it more difficult to treat if it is associated with an infection or a disease. So just keep that in mind when thinking of biofilms. We'll see biofilms later on. We'll see them a lot, especially when we talk about a reproductive infection, associated with like catheters and stuff like that, and stents and heart disease. So we'll see that again. And then the last thing was sporulation. So sporulation, don't forget, I do have a video embedded in this in our lecture. I'm not going to show it on here just so I don't want to get dinged for copyrights on YouTube. So, but I'll talk about it really quick. So what's going to happen is that there are certain bacteria that can go into something that can create something called a spore. And this is like a sort of like a hibernation for them. It's suspended animation. So this is where they kind of just go dormant. And normally they will go dormant whenever the environment is very harsh. So either like extreme heat, like if there's limited nutrients, they will go dormant in order to try to survive. So they'll create something called a spore, which is kind of like a doomsday bunker for them. So what will happen is that the bacteria will sense that the environment is rough and will actually start doing DNA replication. And then the new DNA that is formed is going to be the DNA that is going to go inside the spore. And then the original DNA that they copied from is going to form something called a mother cell. So eventually what is going to happen is that the DNA is going to replicate, as we see right here. So the purple, let me actually zoom in on this guy so we can see it a little bit better. So the purple is going to be the newly replicated DNA. This is what's going to go actually inside the spore at the end. And then this kind of like gray blue, that is the original DNA that they replicated from, and that's going to form something called the mother cell. And then from there, the septum is going to start dividing and everything, and then eventually it will not completely cleave off the cell, but The septum will come completely across, and then the newly replicated DNA and the old DNA is going to become separated. So the new DNA is something called a forespore, which is like a pre-spore cell. And then we have something called the mother cell. And then eventually the mother cell is just going to get degraded and everything. And then the forespore, that's a hard word for me, sorry, will start being enclosed by another membrane in order to create another layer of protection. And like I said, the DNA in the mother cell is going to become degraded, and then that new membrane and everything is going to eventually harden, and it's going to form something called a cortex around that new DNA. And then that cortex layer is just multiple layers of peptidoglycan. It's just a lot of peptidoglycan in there, and then another coat is going to go on the outside of it in order to create another layer of protection. So essentially it's just layers of layers. It's a doomsday bunker for that DNA. And then what's going to happen is that eventually you're going to get a spore. So that's a very, very, very quick generalization of a spore formation. I really just want you to know the idea of why would something create a spore. Not necessarily all the steps, but I would want you to know some basic stuff that I just stated, and that's probably as far as I'll go in terms of sporulation as of right now. So with that, let me go ahead and show you where to find chapter 4 real quick. And of course ACES is down, huh? Let me see if I can try it one more time, guys, real quick before we end our video. Okay, so now it wants to work. Okay, so what you'll do is that you'll go to our course. So we'll go to our giraffe. So let me go to student view so you don't see all this hot mess. Okay, so what you'll do is that you'll go to our announcements, and then you'll see something called the biomolecules worksheet. So under this announcement is where I attached that key. So if you click on the right here, it will pull up the document with the answers on it, so to help with your studying. Okay guys, so that will wrap up our exam lecture review. So I definitely went over quite a bit. It's kind of a long video, sorry. But that is a good place to start when studying, and I hope I gave you some good ideas on what kind of questions are kind of fair game for the exam, and then also how I'm going to format the exam. So how I'm going to format the exam is that it's going to be multiple choice, some free response, a little bit of matching, and complete the table, like complete a table. Nothing too crazy. It's a little, like I said, it's a little bit of everything. Also, don't forget your green Scantrons. So please, please, please make sure you bring your Scantrons to the exam. If y'all have any other questions, definitely email me. And thank you for joining me, and I'll see y'all in class.